So my pleasure to be here. Thanks for the invitation, and it's a pleasure to share some of the results that we do. I understand this is a broad physics audience. I was stressed and emphasized by Alessandro that I should give a broad introduction to the work, so that's what I'll do. So maybe the best way to do that is to start with my own history a little bit, because I was trained as a solid-state physicist working on superconductivity and magnetism and quantum phenomena. So I worked for a while in condensed matter physics and mesoscopic physics, and I moved to sort of biology. And you might think, why would a physicist do that, right? Why would you move from this wonderful world of solid-state physics to biology? And for me, the answer was basically that I got really intrigued by these molecular machines, literal machines. Machines are sort of objects that can basically use some transmission of fuel to do mechanistic actions. And this is actually what happens in biology, because this is a nice cartoon movie based on all crystal structures where you see all these wonderful protein structures. And all these protein structures are sort of, organic machines that do functionality, that make motions, that do, in this particular case, for example, you have this transmembrane protein that does a rotary motion driven by a proton flux generated by the food from your lunch, basically, to generate, to make a local motion here with just an enzymatic action. So it's machines like this which exist in your cells, which got me truly fascinated by biology. And then I was in the 90s already working with nanotechnology. And nanotechnology is just a toolbox. It's just a box of instruments by which you can pick up one such machines, one DNA molecule, a protein molecule, and so forth, and then study these. So most well known in this whole nanotechnology toolbox is Stm Scanning Probes, where you have a small tip going over a surface, and you can image and manipulate surfaces like that. But there's many more tools in the toolbox. One famous one is also the one developed here at NES by Vincent Coquet and David Simon. Is magnetic tweezers. Is another example where you take a simple microscope and you look at a bead which is attached to a DNA molecule. So that's such a simple device. You take a DNA molecule and with some chemistry you attach a bead to that molecule, or at the other end you attach to a surface, and then by having a magnet on top you can pull that bead up and in that way exert a mechanical force on the DNA molecule and measure therefore from measuring actually the position of that bead. With applying a certain force, you can do a sort of stretching force, higher force and then lower force. Basically, the mechanical properties of DNA you can investigate. And more interestingly even, you can take those magnets and then start rotating, and that bead will follow that motion. In doing so, you can basically exert a torque on these DNA molecules and generate these kind of plectonemic supercoil structures. And in doing so, you bring the bead position down as a function of the number of rotations. So here, for example, you see a plot where you see the height of that bead, where we turn up coils to the right or counterclockwise to the left, and we make the position lower. And if we have such a lowered position, now we can flush in some proteins like a topoisomerase. These are proteins that basically grab onto the DNA, make a nick, and in doing so, then, for example, it can swivel around the other the strand that is there, and then basically relax some of the supercoils so you step up. So this is just an example of a measurement where nanotechnology addresses some of the nano world of biology. And I find it quite astonishing because it's a super simple setup where you have an optical microscope, you look at some images of some bead, and you measure the height from that. It's by this simple measurement, you address the action of a single protein on a single DNA molecule. So I think it's very powerful, this whole toolbox of the nanotechnology to address phenomena at this very tiny scale. Yes, oh, please. I would like to have this interactive. So if there are any questions popping up, please interrupt me. Yes?
I didn't, somehow I missed.
Yeah.
How do you twist? How do you do the twisting?
The twisting we do by hand, by having this magnet. By the magnet, basically, we manually start rotating, applying torque, prepare the DNA molecule in a state which has this kind of coils induced, like that. Okay, get that? Now we throw in the enzyme, and then this action occurs as in this cartoon movie, where an enzyme will bind the DNA, cuts one of the two strands, it will swivel about it, religate that strand, and now, since you did something here, the number of coils there have relaxed, and therefore the beat goes up. Get the point? Okay. And so what you measure there is the action of a single protein on a single DNA molecule, which is the point that I was trying to make at this point in the introduction, right? So you see every step here is the action due to a single protein. In our own lab, we've also used another tool of the nanotechnology, which is basically nanostructures. We can, from the whole technology, from silicon technology and microelectronics, there's so many fabrication techniques. And just for fun, I made a compendium here of structures that we made in the past, in the 90s. We made all these kind of small holes or nanowires or grids, and it's all on the same length scale of 100 nanometers. Now I plotted here on the exact same length scale, also 100 nanometer scale bar, applies here as well, some biological structure. Here you see a nuclear pore complex, you see a nuclear lamin, you see ribosomes and so forth. And what you can note is that these structures have the same size scale. So with nanostructures, we can also start addressing single DNA, single protein, single biological structures.
Just
Yes?
a question. I know that the black and white picture is made by electron microscope, but what is the process for the lower picture?
For this, so I'm sorry, this is a cartoon. This is a drawing, a hands-made drawing by David Goodsell, but it's based on crystal structures. So for example, this cartoon is based on the crystal structure of the nuclear pore complex. It's an artist impression, but based on crystal structures known from these proteins. Yes, sorry about that. We get more precise in a minute. So here's my starting point. I was a physicist in the 90s, and we had this toolbox of the nanotechnology, and we wanted to apply this to biology. And you have to realize biology is changing. So classical biology is the biology of the macroscopic world. Here you see Aristotle on the island studying and describing the animal world there, right? And now new technology has opened up, of course, new fields. So one of my fellow men from the same city, actually, Antony van Leeuwenhoek, discovered in the 17th century this microscope. It's a single lens microscope by which he could see all kind of unicellular organisms, what we now call bacteria even, with a magnification up to 500 times or something like that. This is quite amazing. And that opened up the whole world of sort of cell biology. Microbiology was opened up where you could see all this kind of single cell organisms. These are bacteria. This is actually a white blood cell chasing some bacteria in the bloodstream, a very famous movie. All right, and now in this era now, we move into the era of nanobiology. Sometimes people ask me, nanobiology, that sounds a bit like a hypey term, but I think it's very appropriate, just as we like to describe microbiology because that happens to be at the micron scale. Nanobiology is truly nanobiology because at that scale of the few nanometer scale, we have these single proteins, single DNA interacting. And that is where the molecular foundation is for all these biological processes happening in cells. And for those of you a little farther away from biology, just the past half century has opened up a complete world of these protein machines. And this is a cartoon, this is not a real image. Okay, you got that, right? Okay. But it's actually based on the functionality of these structures. Where, for example, here you see this green thing, which is an RNA polymerase, which is opening up the helix, reading off information, and producing this blue molecule, which is a messenger RNA, which is then basically modulated in the nucleus already, and then transported through this nuclear pore that I just showed you a little bit earlier, to another machine, the ribosome, which takes the information from that DNA, encoded in the messenger RNA molecule, and then produces in a complicated way a protein from there. So this is actually the basic process of biology happening in your cells right now. And it's this complexity, and we've learned a lot already from biochemical and bulk molecular biology studies. And now we want to basically, and that's what I've been doing and many others like Vincent and others in the single molecule field, to take these tools from nanotechnology to at the level of single proteins, pick one of those machines out of that cell and study its mechanistic functioning. Right? And at this point in time, we've learned a lot from that, and I'm actually myself also making a next step. I'm also, for the coming 15 years maybe, I want to also do more building with these components. because I'm intrigued, actually, I'm driven in this whole field by this question, what is life? And I don't have a great answer to that yet, which goes much beyond phenomenological description. But in the end, it's just amazing that if you take a living organism, this is a cartoon again, but it's a quite accurate cartoon of an E. coli protein with membrane proteins and cytosolic proteins, and this yellow stuff is the DNA. If you just shake that up and you have it in a test tube, and you just have just a collection of molecules, what is the difference between left and right? I mean, on the right, that object is alive. The collection of molecules on the left is not alive, it's lifeless. What is it in this arrangement of molecules in space and time that makes that living object? And I'm fascinated by that sort of emergence of biological complexity from lifeless components. And at the end of my talk, if time allows, I will say a few words on our first efforts to sort of go in this direction. Just to give a brief overview, my own group works in a number of areas. We work on single molecule biophysics, basically this toolbox that we do stretching and bending and measuring on single DNA molecules, DNA-protein interactions. We do a lot of work on solid-state nanopores, very tiny holes to which we traverse molecules like DNA and develop sequencing technology based on that. And we do now also quite a bit of work on nanostructures to confine single bacteria. I cannot cover all the work, so what I basically decided to do, to give you two examples from recent work from my lab. One is basically something to do with this supercoiling structure of DNA, and the second part of my talk will be about using these nanofabricated structures to confine bacteria and learn about their division properties and their properties. So first, that first part of the talk, that's actually work done by a postdoc in my lab, Sang-Yoon Kim and Maipal Ganji, in collaboration with Eloy Bonicieri's lab at Delft as well. And it addresses the question about the genetic information, whether that can also provide sort of the information about the mechanical structure of DNA. Now, again, about that supercoiling, you can start playing, as I did already a little bit, with sort of twisting wires like that, right, and making this kind of plectinemes. If you first twist it, you just basically store twist in the molecule. If you twist it further, you start making what we call plectinemes, plectinemic supercoils. That's a topological structure, as you can see here on the right. Normally, DNA, of course, with the famous double helix structure, is already twisted intrinsically. You can over-twist it or under-twist it, and you can create these plectineme structures. This kind of structure is not just only a physicist playground. It's biologically crucial. It's vital for cells. If you don't have the control over the exact amount of plectinemic supercoils, cells die. So, and that's because it's involved in regulating many processes, the compaction of DNA. In each of your cells, you have like 2 meters of DNA in each of your cells. And that 2 meters of DNA is condensed into a structure of a few microns in the nucleus of your cell. So in that condensation, this supercoiling plays an important role. But it's not only that, it's also in the transcription, the replication, and these kind of processes. Now, the DNA is supercoiled in your cells, but it's organized in topological domains of, say, tens or hundreds of kb, tens of microns of DNA, basically make one domain. And within that domain, you basically have then the stuff supercoiled. Now, you have a different polarity, if you like. So you can do positive twist or negative twist stored in this kind of thing. Most of your DNA is sort of negatively, it's a bit underwound, negatively supercoiled. A bit, the normal twist of the DNA is slightly underwound, or relaxed a little bit. But some of the parts of the DNA are actually overwound. And that has to do with the biological activity. The stuff that actually needs to be transcribed, read out for the genetic information is actually slightly underwound. The stuff that you want to store, because in your skin cells you need different types of genetic information in your heart cell, your brain cells, and so forth, that actually is slightly overwound. So there's actually a distribution along the genome of this kind of supercoiling. Also, you have to realize that the supercoiling is not a static given thing, but it's completely rearranged all the time, dynamically. So, for example, is one of those machines, the RNA polymerase, which reads out the genetic information, walks along the DNA to basically read off the information, it spools in that helical filament, and therefore it generates supercoils ahead of it and in the wake of it, of the different polarities. So this rearranges and this is relaxed by topolarisomases and other proteins and so forth. Now, people have imaged this in the past. People in the 90s in particular, people have done electron microscopy studies of this kind of supercoiling structure. But there's so many questions which are unanswered which have to relate with the dynamics. How are these dynamically changed? What is the dynamics of this stuff? How is it relating to genome sequence and so forth? A couple of years ago, oh, this slide doesn't work. A couple of years ago, we basically made a variant of this magnetic tweezers where we do not simply have the magnets on top, so we pull up a bead on top, but we actually slide them to the side. And for that reason, we could put the DNA in the imaging plane, right? So this basically can be done like that, and then you pull this to the right. And what you see here is the density of DNA along a molecule. So the spots of high intensity are spots where there's one of those plectinemes. And you can see now the dynamics. So we can actually coil up these molecules and see them diffuse. So they slowly move. They have the random diffusion properties moving along the DNA molecule. They can also hop. We discovered they can hop by basically terminating a new one. And certainly over microns, you vanish one and you actually generate a second one, microns away. So there's very interesting dynamics. This was already published a couple of years ago, so I will not focus on that in this talk, because in this talk I want to ask a question on something that we are currently working on in the lab. It's not yet published, but it's a very interesting question. Is there actually in the genetic code also information about that mechanical structure? Can the DNA sequence spin the position to have him preferably at some sites of the DNA, or basically push him away from other sites in the DNA? And for addressing that, actually, we made a couple of technical developments first. One is this technique for the side pulling of the magnetic tweezers is very nice, but it's very laborious. So we found a new technique where we actually inserted basically a new fluorophore which did not nick the DNA, which is basically an intercalating fluorophore, cytox orange, which basically intercalates between the bases of the DNA and changes the twist a little bit. So what we do in our new technique is to basically have a DNA molecule, attach it in a flow to a surface, stretch out the molecule with the flow, attach the other part to the surface as well. And once we've done that, we made this construct. We flush in the cytox orange intercalator, which self-intercalates into the DNA molecule and induces, for that reason, because of the different twist, induces the plectinemes. So what you can see here on the right is a wide field view where you can see multiple molecules which have these bright spots moving back and forth on the DNA. So this is nice because it's now a high throughput technique to directly just visualize the plectinemes on many molecules. And then we can do some data analysis, of course, where we can take a chymograph. Basically, this is time, and this is position. So we can trace the position of these blobs as a function of time. We can actually transfer that position in microns to position in kilobases along the stretched out molecule, because we realized that a very bright spot takes up a piece of DNA where you have a start and tip and the end of the plectineme structure, so we can stretch it out in kb. And from those positions along the positioning in kilobase along the molecule, we can project all that to make a density. So that's the plectineme density. The chance that you have a plectineme located at a certain point along this linear stretched out DNA molecule. Right? Again, if there's any questions, please interrupt me. So we want to ask that question. I'm going to show you some data on these plectineme densities. And I'm going to show you something, and we'll discuss a bit the mechanism. Why does it pin at certain spots? The first thing is, the basic thing is that different DNA sequences give us different plectineme densities. Here you see a typical DNA molecule. What I plotted here is the GC contents. As you know, the DNA is built from GC and AT base pairs. So some areas have more GC on average, and other areas have more AT on average. So on a typical molecule that fluctuates quite a bit, and you see a certain profile of the plectinemes. And here we engineered a molecule which actually has a relatively low fluctuation in the GC content. Well, and we find something else. So the first thing to note is that different DNA profiles give you different densities of the plectineme density, right? Of the plectineme occurrence. So one thing, you might ask, why is it different, right? And so we guessed it has to do with this AT-GC content. So we constructed a number of these molecules where we have a high AT region, so low GC, high AT, in a high GC background. And we made that longer and longer and longer. We started with 250 bases in a row, up to 4,000 in a row. And lo and behold, you saw a very strong peak emerging if you make this large AT region. So apparently this pinning correlates with AT regions of DNA. Okay? But that's not the full story, because we saw, for example, also this pronounced peak at a region which doesn't have a strong AT region at all. So what is that? So we did look into that a bit more detail. So we took that peak, we copied it with just molecular biology techniques to the center of the molecule. And yes, we found a peak there as well. So that relates quite nicely. What is special about that? We looked for the sequence in there. We thought maybe it's these poly A regions, which means like A in the sequence on a row, which has been discussed in the literature to make intrinsically curved regions of the DNA. So maybe it's that. So what we did, we mutated it that way. So we replaced some of these A bases by G bases, for example. And indeed, the peak vanished. And that's kind of nice. But then we replaced it by T molecules. And of course, A pairs with T. So actually, it's just AT, TA, and so forth. And then the peak resurfaced. So we thought maybe it's just the presence of AT base pairs rather than poly A tracks there. But then we got even more confused because we tested and tested more controls. And for example, we shuffled these AT track regions to other positions. And then basically the peak would vanish. So we're actually quite puzzled at some point. We clearly see a sort of DNA sequence dependence. And the exact origin of that has to relate with AT base pairs, which is quite subtle. So we started thinking about the mechanism. And you have to realize cytoplactinine is this object. What pins it particularly important is this tip, because that tip is where the DNA is bound at the end. So if I make one of those plactinines, of course, there's a stem and a tip. Of course, there's some energy stored in mechanically deforming this part, but this tip is the major part because you need a mechanical bending energy to bend that DNA, which is a high energy. To bend a straight piece of DNA over this range is like 25 kT or so. It's a lot of thermal energy to bend that over the relevant range in a sort of tens of base pairs. So what can cause this pinning? So we speculate that the pinning has to do with the sequence at the very end. It might be that high AT regions sort of make a local melting, and then basically that band would collapse into a complete sort of collapsed structure which is demelted here. That's one proposal. We think in the end it's not very obvious that it's unlikely that this explains our data, but I don't have time. You can ask me about it later.
Sorry.
Yeah, go ahead. Yes, please.
Very quick question. You showed the density of these plactinines. Does that incorporate also because these plactinines are also elongated? Do you take that into account?
So when I show the density, I basically, because the conversion we did from microns to kilobases, what I'm plotting there is the position of the tip.
Okay, so regardless of how big
we see DNA density, which we basically then project into plactinine, the position of the plactinine. And indeed, if we find like a 3 kb long plactinine, we take the center of that structure as the position of the plactinine. Right. Yeah. Yes.
Sorry. Give me this. If I take a multicellular organism, is it clear that it's the same DNA everywhere?
Yep.
But is it clear that the same plactinine everywhere, or does it depend on which tissues they are involved and which organ?
So
is it specific?
So in our cells, we have hundreds of different types of cells, and their differentiation is governed by many factors. But part of the DNA is used in, different parts of DNA are used in different cells. Some are used in all cells, but some are used in specific cells. And indeed, the whole pattern of what is expressed in transcription is different in different cells, and that's also regulated, but that's a new speculative, that's up for discussion. We think also the supercoiling structure relates to this. But that's a new notion, that's not conventional wisdom, that's actually one of the proposals coming out of this type of research. There's also many other proteins involved in packaging this DNA. That's, of course, also a very important role. Yeah. But you're quite right. So we're basically we're thinking about the mechanism. What is pinning these plactinines? And we're thinking actually this intrinsic curvature may play an important role. And let me explain that to you. So what we do is that we take some classic models for DNA. You can look at the DNA, and from base pair to base pair, you can characterize the exact mechanical arrangement of how this next base pair is positioned. So I take a base pair, place the next base pair on top of it, and depending on what base pair it is, this one and this one, there will be a certain roll angle and a tilt angle and so forth, right? And if you take these roll and tilt angles as a wedge angle, the different base pairs, like AA to AC, AC, AG, and so forth, will have slightly different angles. So what we now can do is to take that 20,000 long base pair long molecule and to build a mechanical model like that. This is now only the equilibrium structure, right? No thermal fluctuations. And if we do so, we get a sort of structure where locally sometimes you happen to have certain curvatures. Okay? Now, interestingly, if you think about it, if you look at the theory for plectoniums developed by John Marco and Sebastian Neukirch, you can look at these plectoniums, at these tips. These tips are very similar to what we call curls, the first entry of such a tip. And basically, if you look at the top half of that, it's exactly what I just was telling you. That's NR parameters, like a few piconewtons, positive supercoiling. You have to bend that DNA 180 degrees over a distance of sort of like 30 base pairs, 32 base pairs. Okay? That's an energy, if you start out straight, that's an energy of 25 kT. However, if you have a region of a DNA where it's already intrinsically curved, you can start here and bend only a little bit further to get to the 180 degrees. And then you have to pay much less mechanical energy to get there. So that's interesting. So you have a simple expression for the bending energy, which is just the mechanical expression for the bending energy, which if you have an intrinsic curvature, this energy is much lower. You pay maybe 10 kT or 15 kT. Now, if this ends up in a Boltzmann factor for the propensity to bend at a certain point, you can calculate at every position along the whole genome what is the energy you have to pay, so the propensity to make a plectinin tip at that point. If we then convolute that with the blue curve to our experimental resolution of a few kb, we get curves actually from this model that predict, basically based on the sequence, where the plectiniums were actually pinned. And interestingly, this compares quite nice to our experimental results, at least qualitatively. So this is some data from the model. No fit parameters, right? Just put in the sequence, put in this mechanical model, and this is what we get for the plectinin density. So what you see here is that end peak that I was talking about directly comes out of the calculated results, which is very gratifying. And furthermore, the thing that I showed you is the AT-rich peak. AT-rich region would give you a peak, and if you shuffle it down, shuffle it around, it would not give you a peak. So it seems to work quite nicely as a starting point. This is sort of where we are in modeling. We've now actually even made predictions of certain sequences that actually would predict to give a curvature, made them in reality, and lo and behold, we got peaks there. So we're quite happy. We're looking now at E. coli genome and just the genome of real organisms to extract certain regions, which gives you this high curved region and see if these plectiniums spin. So what we conclude from this part of the talk is where we have a nanotechnology technique to address molecules at the nanoscale, and we learn about these basic properties, and we learn basically that there's a sequence dependence for this plectinin pinning. And the broader significance of that is that we think of DNA as the genomic carrier, and rightly so, because it carries the information needed for making all this wonderful complexity in biology. But there's actually another layer of information maybe encoded in the genome, which is sort of where these plectiniums are preferred to be. And that's interesting. So there may be a mechanical information stored into this genetic information from the genome. Let me go to the second part of my talk, unless there's a particular question on this particular data. All right, so the second part, I switch gears a little bit because we do a lot of single molecule biophysics, biophysics at the level of single DNA protein molecules. Now I want to basically change gears to single cells. So this part of the talk is maybe about pattern formation of cell division proteins and about bacterial shape. I got to actually study bacteria in nanostructures because of an interest in evolution and ecology. And in real evolution ecology, people do experiments like these, for example, where they chop down parts of woods. This is in Carolina. And then they study the spreading of a certain shrub. People are interested in the population dynamics and the sort of ecology of spreading species of shrub and competition and things like that. We wanted to do the same with bacteria. So we make microfabricated structures, kind of boxes of 100 micron wide. And we put these chambers and we put bacteria in there. And we have them connect between this kind of channels so they can swim from one chamber to the next. And we give some chambers a lot of food and others low food. And we do sort of ecological games like that. So we fill them with bacteria, which is fluorescent, so we can see them in the microscope. And they swim around nicely. And we do all kind of research with that. We did some ecological experiments with that. But I will, for this talk, focus on the biophysics, which is very interesting. So one of the things we discovered is actually that these E. coli bacteria can go through very tiny slits. This is something weird that we discovered. And that's the following, came from a simple control experiment. I showed you these chambers and sort of channels connecting them. This is a side view of such a channel. Look at the scale, but like a micron. This is an E. coli. Normal E. coli bacteria, this is a sort of model organism, a bacterium, lives in your intestines. It's like a micron wide and a few microns long. Okay? So this is similar size. Indeed, if we make the slit a little bit wider, we can see E. coli bacteria swim nicely from one chamber to the next. And we can measure the swimming speed, 20 microns a second. And if we make the channel smaller and smaller and smaller, at 1 micron size, it stops swimming. End of the boring control experiment. But that was not really the end of the control experiment, because when we looked for a few hours, we left them in there, then actually what we saw is that basically these bacteria would still penetrate that slit, even if that slit was only like 400 nanometers wide. So even a 400 nanometer wide slit would be 2.5 times as small as the width of the bacterium. So it would be sort of squeezed like a pancake and still be in there, grow, divide, grow, divide, grow, divide. And indeed, we made a different layout where we could image these kind of things, so not very deep and narrow, but very wide and shallow, so we can have them in the imaging plane. And what we would see here is the bacteria have very weird shapes. This is the normal E. coli shape, 1 micron across and a few microns long. This would be pancake-shaped bacteria, which are sort of 5 micron wide and only like 0.4 micron high. Okay, and so that's okay, that maybe you can squeeze bacteria. But what was amazing to us is that they would grow and divide. So division in general is a very regulated process. So how can it be that they so accurately still divide? Because normal bacteria grow and divide like this. So you have this bacteria, but grows longer, longer, longer. Then it pinches off in the center, and you have two daughter bacteria. These ones would sort of expand, invaginate at one side, and then the sort of division plane would appear. And so they're very plastic. And this may be very relevant because so much bacteria are in soil where you have micron cavities and all that, and they can penetrate that. So this got me really interested in bacterial cell division. And we did a number of studies in that area. I'm going to show you some of that. So as I said, these bacteria, they grow, they expand their length. Then there's a so-called Z-ring placed at the center, actually precisely at the center. It's regulated by proteins to put it precisely at the center, not a little bit 20% off or something like that, exactly within a percent at cell center. And then that Z-ring starts to constrict until we have two daughter cells. Well, this Z-ring is actually underlied by a protein called FDSZ, which is linked to the membrane by other proteins. There's many accessory proteins helping to organize this whole process. But this is the main player. The placement is done, as I said, very precisely at mid-cell, and that's regulated by two mechanisms. One is called the MINT proteins. I'm a physicist in biology. I'm amazed by this. This is how nature regulates the centering of your protein machinery at cell center, which is done like this, where you have certain proteins that all assemble on one cap, then they'll all let go. They all go to the other cap and assemble there. Then they all let go, they come back, and they oscillate back and forth. And they are an antagonist to the cell division machinery. So wherever there's a high density, average density of these proteins, the cell machinery will not be. So you see these oscillations of these, and then the cell division machinery is basically assembled at the center where the density of these proteins is low. I find it very fascinating and unexpected way to get the center of the cell. Another mechanism for that is nucleoid occlusion. So of course these cells have DNA. They copy their DNA. This DNA segregates into two copies of the DNA, and then there's a slightly lower density of the DNA in the cell center, and that actually drives the center ring also to cell center as well. And if we found out there's these flat bacteria, they have multiple copies of the DNA. We found actually this latter mechanism that drives this particular cell division. And we could do anticorrelations of this nucleoid in red and the Z ring, actually the Z arc. So the picture that emerges is actually the nucleus open up gaps. That's where the cell division machinery then is assembling. And then like a sort of knife, it makes its way sorry about that. It makes its way through the cell body, separating the cells into the daughter cells. So this really got me fascinated in cell division. So this is a field with a sort of decade-long history, bacterial cell division. It's still a hot topic because there's so many questions. It's actually 25 years ago that people discovered that this ring is sort of the governing principle of the cell division in E. coli. And yet there's many, many questions about the structure, about the dynamics of that. I just want to also tell a story, which I'm sorry, this slide is sort of messed up. My picture's still there, but all my important collaborators are not there, as well as the important question. I don't know why this is. This is a PowerPoint problem. Let's live with it. I'm sorry about that. The point of what we're trying to make is that we just published a paper in Science 1 month ago, and this was an important collaboration with Ethan Gardner from Harvard, Yves Brun from Indiana, Seamus Holden, and myself. And the question there that we addressed is actually what's the mechanism of this dynamics of this FDSZ ring? And that's quite interesting. So from Yves Brun's work, what we could do is to image actually in cross-section this E. coli. This actually is done in Bacillus. It's a different bacterium. But, okay, so Bacillus subtilis. You build that, basically that cell wall in the center, starting from the outside to inside. With different colors, we could color code the cell wall that's being assembled there from the outside to the inside. And you can see the blue, the green, and the red being assembled from outside to inside. But what was interesting and unexpected is that actually this cell wall would not be sort of built everywhere homogeneously a little bit and then going from the outside to the inside, but it would go in patches. And actually, if you would do 2 patches behind it, first in blue, green and then in red, actually these patches would be always neighbors from each other, like that, suggesting that it's sort of like built sequentially along the cell circumference. And indeed, we found that these FDSZ filaments, they move along the circumference of this thing, of this bacterium. They're not a static object, but they move. Because this was measured from a turf microscopy, so you take these bacteria and you measure only the bottom part, and then you can see through your field of view in turf that these bacteria, this machinery is passing by through your optical field. Okay, and what we did actually from Delft with all our nanofabrication stuff is to make a different layout where we put bacteria standing upside down, because normally many people work on bacteria and they lie on the glass slide like that. We will put them upside down like that, which is very convenient because then you can measure in the imaging plane this ring, and you can get pictures like this. On the short time scale, on the long time scale, you can see this large ring sort of slowly contracting to small rings like that, which is kind of nice. And from chymographs, then you can see lines of this kind of protrusions there going along the ring, which are just straight lines showing that there's objects walking along the circumference of this bacterium. Now, what was interesting, that we could relate this FDSZ motion to actually the machine, the protein machine, which is building the cell wall. And that would show exactly the same type of dynamics. Again, in this turf mode, we would see the PBP synthase. That's a word. That's the name of this machine that builds cell wall, which would run this exact same speed as the FDSZ. And so the picture that emerges actually from this FDSZ is that it's proceeding. And it's proceeding by a particular mechanism called treadmilling. Now, treadmilling is, I don't know whether you're familiar with that, that's basically the building of a polymer at one end and the breaking off of the polymer at the other end. And if you look at this filament, it's clearly as a filament moving from left to right. But if you look at any one monomer, it's actually static. This one stays there all the time while the filament moves to the right. This is treadmilling, and this is what we see for the FDSZ, because if you label individual FDSZ monomers, they're static in the chymographs. This is kind of interesting. So the picture that emerges from this is actually this one, where we have these treadmilling FDSZ filaments in red here, which are correlated with the motion of this cell wall, which is actually building more and more cell wall, and slowly from the outside to the inside, it's building more and more cell wall in making the septum and therefore providing the cell division machinery. So this was a study, it's an interesting study, I think, on the mechanism of this cell wall, cell division machinery. I have many more things that I can show. I have to look a little bit on what kind of thing I'm going to present to you. Let me try this one. This is actually a further study of these MIN proteins. I told you that we have these proteins that make the cap on the bacterium. This is a cross-section of the bacterium. Make a cap here. They cover the cap in green here. And there's another protein that kicks it off, brings it back to the cytosol, and starts building a cap on the other side. So this is an example. This is all driven by protein membrane interactions. So there are proteins here in green that bind to the membrane, and there's another protein in the red that binds to that green protein, kicks it off. So there's three components. There's the min-D and the min-E protein, and there's ATP, which is the fuel, which is basically hydrolyzed to get basically ADP and phosphate. And this is a simple interaction between two components, where one is actually self-enhancing because it's cooperative binding of this protein to the membrane, and the other one is inhibiting. And this is an example of a Turing pattern. Alan Turing in 1952 wrote a paper at the end of his life about shape and form in biology, morphogenesis. And actually, he predicted these reaction diffusion systems. Systems where you just have the reaction of multiple components and diffusion of that, which underlies all these wonderful complex patterns in biology. The stripes of a zebra basically derive of that. I mean, basically the patterns in Drosophila embryo and so forth. It's driven by this reaction of a few components and diffusion. So it's a wonderful thing because it's just a few components giving you this interesting spatial patterns in biology. And this example, what I just showed you by these min protein oscillations, is an example of that at the cellular level. So within one cell, even such patterns actually play a role to organize proteins within a cell in a spatial way. Now, what we did is to study this, a very talented student, Fabai Wu, did this work in my lab, is to take E. coli cells and now to look at the effect of spatial boundaries on these Turing patterns. So what we did is to take cells, to mold them a little bit, in the sense of do some antibiotics which basically soften their shape, so they will expand. And we had another antibiotic to prevent the cell division. And then they would grow, and then we would put them actually in nanofabricated shapes. So in doing so, we could, for example, make a triangular shape or a rectangle or something, and in the course of a few hours, grow these cells to these shapes, and then investigate these min patterns, these oscillations. So we can probe these Turing patterns, but now in novel shapes. So understand more from a physicist's point of view what determines these patterns and how the bacterial shape is sort of regulating these patterns so that the cell division machinery can do its work. So that's what we did, and we can do wonderful things like spell TU Delft and things like that. Which is still sort of interesting because this letter F, for example, is one cell. It's a living cell. And actually in green, you see the spatial pattern of the proteins doing their dance within this one cell. And we can study the effect of shape and size. So for example, we take square cells, which go from 2 by 2 by 1. No more squeezing, by the way, they're all 1 micron high. 2 by 2 by 1 to 8 by 8 by 1 micron. And we can basically see the patterns of these min proteins in these cells. So we can see rotational patterns, diagonal patterns here, side-to-side oscillations, or higher order diagonal patterns, and so forth. And they're the same shape, but very different patterns, actually, so that's one thing to note. We can quantify that, and we can quantify a typical size. It's sort of characteristic size of 4 or 5 microns, which fits to the side of the thing. It will make a side-to-side oscillation. If you make it smaller, that same size goes along the diagonal. That makes a sort of intuitive sense, right? It's a sort of characteristic size that fits the pattern to the shape of the structure. We get also different shapes, like rectangular cells. We see yet other patterns. For example, higher order modes, if you make very long cells, a bit like filamentous cells. But also, interestingly, transversal modes. So for most of these patterns, you see a sort of the long direction picks up the oscillation side to side, like a pole-to-pole oscillation. But these cells, actually, which have a width of about 5 microns, they make traversal oscillations. And indeed, there are some organisms that do the cell division not by growing longer and pinching off in the center, but by growing wider and pinching off from head to tail, like that. And they're probably underlied by these kind of patterns. So shape is important to this pattern. So what we learned from this is actually that these proteins sense the symmetry axis of the system. Because actually, if we look at square cells, we see diagonal patterns and these pole-to-pole patterns. Whereas in the rectangles, if you deviate a little bit from square shape, you lose this diagonal as a symmetry axis. And indeed, they do not show this kind of diagonal patterns anymore. And so we learn actually, and that's actually counter other models in the literature. Other people would say, oh, the system always picks up the longest length scale, but it's not, because the longest length is the diagonal, but it picks up the symmetry axis of the system. So we make a phase diagram and show which patterns are robust in what way and so forth. Also interestingly, we can actually do a modeling. This is work by Erwin Frey, and other people have developed models. And we can take the sort of microscopic models like this. Every arrow here is, of course, a biochemical rate in the process, the binding of a protein, the binding of another protein, and so forth. If we can take this one set of arrows, with one set of biochemical rates, we can actually recapitulate all these patterns in a model. And actually, we can use these patterns to tune some of these rates. And we found actually this cooperative binding is very important. So from a global fit of these patterns to what we experimentally observe, we can learn about sort of biochemical internal structures there. So that's kind of interesting. So we learned that we can sculpt these live bacteria with nanofabrication tools. We can see that the min proteins sense the symmetry and scale. Now we expanded this recently to also study chromatin structure, the structure of chromosomes in cells. So there we actually added one more trick. Because normally if your cell grows, you start replicating the DNA to a second copy of the DNA and so forth, right? If we prevent that process by halting the replication early phase, we can actually have one chromosome but still have the cell grow. So we have an expanding cytosol and only one chromosome in there, which is giving us a complete new window on imaging the chromosome, because normally that chromosome is confined in this enormously squeezed volume of a tiny cell. Now we can relax the volume of the cell and start actually seeing the circular shape of the E. coli genome. And indeed, this is a direct visualization of the circular genome. People have done molecular biology tricks, it's called high-C techniques, to sort of correlate which part of the DNA correlates with other parts. But now we can do a direct visualization and do that actually in real space. So we can image the genome as a function of its position along the genomic coordinates and see interesting structure where, say, the TER region is very dilute and other regions are more dense and so forth. Not only that, we can do dynamics. We can actually see this rearranged on the timescale of tens of seconds and sort of dynamically see how this chromosomal mass rearranges and what area is determining that. Now, in the last few minutes, maybe show you what I promised you, sort of my dream for the future. Can we build this kind of biological complexity from the bottom up? Biology is hugely complex, so this is a truly ambitious project, but I'm still quite interested in it. So my question would be, can we bottom up build a cell? Can we take a vesicle, fill it with purified proteins, and build up, you might call build up a bottom-up biology? Build a simple, complex systems from components. And from my reductionist physicist point of view, I would like to build systems that I understand and build up more and more complexity from there. So we do this in various ways. One way is to build what we call in vitro systems. It's basically a nanofabricated chamber which we coat with a lipid, which mimics a little bit the inside of a cell. Then fill it with DNA and proteins and study this kind of thing and have some valves to close off, worked by my postdoc Jaron Kaspi. Where, for example, I showed you some of these min oscillations. Here's an example of small chambers which we filled with min D protein, min E, and ATP, and you see these oscillations occurring, right? You see in this rectangular cells these oscillations occurring, showing that we can build this kind of in vitro systems indeed for these min proteins, for example. And then we did again the phase diagram, different dimensions give you different patterns, and spirals seem to be actually the major pattern there. And actually the longitudinal pole oscillation is a derived phenomena from the spirals. And we can have some phase diagram. The second approach there is to build more cell-like objects rather than nanofabricated chambers in silicon or glass. And actually what my dream is to basically build this kind of vesicles, to fill them with DNA and proteins, build such a ring system, place that at cell center, modulated again by proteins or DNA, and then have that constrict into daughter cells. That's sort of my research program for the coming 10 years. And here's what we are starting to do. So we made a new technique to build liposomes on chip, because I really want to do this on a chip where I can manipulate single vesicles and image them and so forth. So we have a new technique where we have like a water flow from the top and the bottom, and an inner water flow in blue here, and in red here two octanol octanol fluid containing lipids. And then basically you form a double emulsion where you have a liposome and an octanol droplet which spontaneously buds off after a few minutes. So after a few minutes we are left with perfect liposomes filled with the liquid from the inner aqueous solution. This is well known from droplets, water in oil. There's many people doing this, but to get through liposomes, actually, it's a bit more delicate and interesting we have new technique to do that actually now. We are working on better and better structures, getting them purified, get rid of those octanol droplets by simply density purification, worked by Sira Despan, a postdoc in my lab. And now we're starting to make the very first steps in the direction of making these rings, worked by Fede van der Lista, where we fill basically these same liposomes now with these FDSZ proteins, and we're starting to make ring structures. As you can see, one of the ring structures in the liposome here, and try to get some action in contraction and so on. This is sort of where we are. This is a status update of where we are with this project. So what I showed you in the second part of this talk is that we can use our microfabrication, nanofabrication techniques to address new phenomena at the single bacterial cell level, and basically going in the direction of even synthetic systems which may build cells in the long run. So the bottom line basically of this whole talk is that all these nanotechnology tools are an interesting toolbox to address biology at the single molecule, the single cell level. And I'm happy to take more questions, and I thank my group members, and thank you for your attention.
I have a very obvious question. It's a fact that you never show in any of your picture the temperature at which you work, and it's visible that if I lower the temperature, it will be completely frozen. So when you give a time scale, which is very precise, you must say at the same time, are you working at room temperature of 21 C or at 37 C or something like that?
It's a good point. I should add this. Most experiments that I showed were done at room temperature. Some of the experiments on bacteria are done at 37 degrees. Yeah, so that's the range sort of where we did the temperature. And of course, temperature comes in as an important parameter because you have to control that. For example, you cannot do your bacteria experiments at 60 degrees because the bacteria will die or something. And you cannot do it at minus 10 degrees either because they freeze. Yeah, that's obvious, yeah.
Sorry, I'm not sure my question makes any sense, but
give it a try.
Why does E. coli goes out of its way to squeeze itself into this narrow tube? I mean,
that's a very relevant question. So the answer is the following. We have these chambers, and these E. coli thriving in there, and they're dividing. It gets very busy as in high population density, and they get less and less food because of the high population density. And now this slit actually is connecting to this other chamber, which still has plenty of food. So it's partly driven by chemotaxis. What does mean? They sense the chemistry. They smell, and they go where the food is. So they really have an urge to go there, and they mechanically dock then, and they grow in that direction.
I have a sort of a follow-up, which occurred to me while you were talking, but I didn't want to interrupt. I'm not sure. Maybe you said this, but I did not see it. What is the slowdown of this crawl through the slit, and how does it depend on the slit size? I mean, you were saying it's hours, right?
Yeah.
The control was quite fast. What was it, minutes or something? The control experiment where it was at least as big as the bacteria.
Oh, that's seconds actually.
That's seconds.
Yeah.
Okay. And then
100 micron, 20 micron per second, 5 seconds.
Right. And then it turns into hours when you.
Yes.
And what about cell division?
Okay. So there's many questions here. So the normal swimming speed of bacteria is like 20 microns a second or so. 20 microns per second. So they can swim happily over a couple of hundred microns or so in second scale. In these other things, they could not swim anymore. Then they swim with a flagella. So they propel themselves with a flagella forward. And now in this other confined space, they cannot swim anymore. They can run the flagella, but nothing will happen. They really are there, they're confined, and they simply grow. And then they divide, as I was telling you, sort of that's an amazing thing, but they do. So you can image that. They grow, divide, and they grow, divide. Now, I think it's just mutual pressure. They're pressing themselves to the right, and that's not slowing down, actually. Actually, it's quite regularly going forward. And that's basically just going on continuously until they reach the other chamber. And then people ask also, what happens then? Then they come out, they pop out. And then they're slightly defective in their shape. They're a little bulgy, blobby. And that's because they basically accumulated cell wall defects. Normally, E. coli has an inner membrane, a cell wall for peptide glycan, some sugar, and another membrane. In this process, while they live in this slit, this peptidoglycan layer is a little defective. They come out, they have some bulges, but the genetic program is still the same. Actually, we checked that, they even sequenced the stuff that came out, and it was the same sequence. It didn't evolve in that sense. And they come out, and they still have a program for that cylindrical cell wall, and they start budding off some parts, and then they basically, after 2-3 generations, they're back to normal rod shape, and they basically explode in their population, and they fill up the whole thing with these rod-shaped bacteria again.
I didn't fully understand. So, are you saying that they are not really are they crawling?
I don't think
through
I don't think they're actively crawling. I think they're basically pushed. They grow, and just by having stuff here there, and they grow like that.
So the speed of penetration basically is dictated by the growth
Yeah, growth and division. Growth more than division, actually. Division is just taking the same biomass, making an extra wall there. But growth, yeah, it's setting by growth. Yeah, correct.
So I think we can thank again Professor Dekker for the
You're welcome.